All right, so our next chapter is the infectious cycle. So what we're going to talk about, this will basically give you, so first we got the very initial idea about the world of viruses in the first chapter. That's an introduction. Here, we are going to see that this will give you an overview of the process in some of the initial slide. And then it will also tell you that how in the infectious cycle process, how we enumerate or measure the amount of viruses in an infectious process. So these are the two main parts of this talk. So what we are looking at, and we will be looking at these pictures all the time, like these cartoons, that when a virus actually infects a cell, we see that there is an eclipse period going on. At that time, a lot of different things are happening after the virus enters into the cell. And many of these things are happening simultaneously at the same concerted way. But just for our understanding, we actually make them as discrete processes so that it's easy to understand. That's basically what it is. So just giving an example in that. Say for an example, you have a cell right here. What you are seeing, you have a nucleus, and you have the first step is what you call as an attachment and entry. So the virus has to bind to a cognate receptor, and it will be taken into the cell. Once it goes into the cell, it would release its nucleic acid, and that will be translated by the ribosome. And along with that, when it is translating its ribosome into the different proteins, also it will replicate its genome, which needs to be packaged inside the virus. Remember that, again, that when you have the virus, we talked about that the virus is just like a box, like this. So this box, this is made up of protein. This is protein. And then inside that, you will have some sort of nucleic acid, either DNA or RNA. And that is your genome of the virus. So basically, once it goes inside the cell, you have to do two things. You have to make the proteins, which will make the box. And also you have to replicate the genome that will be packaged inside that box. So you need to do this function. So to do the first function, you need a translation, which is making all the proteins which are needed for making the box. And also you need to replicate the genome. Once all these proteins that what you are seeing right here, VP0, VP2, VP1, these are the proteins. And we're going to talk about this is poliovirus. They are, many of these are processed and we're going to talk about. But once these are made, inside them, the genome is packaged, and this process is called the assembly. So you can see that from the translation, genome replication, assembly, this is the eclipse period. All these things are happening in the eclipse period. Once the whole thing is assembled, it will just make the cell burst open and come out, or they will bud out one at a time, eventually killing the cell. All right, so what do you need to study an infectious cell? And again, very important thing is like it's too bold, but I also is a live cell. But any cell? Nope. What kind of cell? And again, remember that we are talking about a host and its cell. And yes, of course, in the laboratory, we can just do the cell culture, but you need a cell replicate, a specific type of cell from the specific host the virus infect. We just talked the other day that the plant viruses don't even infect us. They just pass through it. Insect viruses don't infect us, or the bacteriophages of the bacteria don't infect us. So here are some very important definition and very important concepts. So make sure that in an extra important slide. So a susceptible cell has the functional receptor for the given virus. The cell may or may not be able to support viral replication. So what does that mean? That means that if by a mechanical means this cell, the susceptible cell, the virus touches or binds to its receptor, the cell will take the virus inside. But that can do two things. it may not even replicate the virus if it is not a right kind of cell. So just remember that this is an important concept. It does not mean if the virus can enter the cell, does not mean that it may replicate. It may, of course, it may replicate, and we'll see that definition, but it may not replicate the virus either. So that's an important. Susceptible means it can just enter the cell, that's it. On the other hand, a resistant cell has no receptor, and it may or may not be competent to support viral replication. Now here, you just probably puzzled that you're saying, okay, all right, the virus cannot enter the cell, and so it is not going to replicate the virus. How it is going to, you are saying that they may or may not be competent to support the replication. So how it can replicate? This is the thing that you just need to understand here. This is the main concept here. There is a process, this is a typical molecular biology technique where you take the virus and you package into a lipid nanoparticle, which is a liposome. This is exactly the way the Pfizer and the Moderna vaccine basically work. But remember that they do not contain a virus, they contain the mRNA. Entirely different. But what you do is that you take a lipid particle, put the virus inside, and these are commercially available. It's called a lipofectamine. You just buy it from Ginkgo-Biotech and that. You put the virus, you take them, mix them, and you will have lipid nanoparticles enclosing the virus. And what happens that when you mix it with the cell, this lipid nanoparticle, this outer membrane, is going to fuse with that resistant cell, and the virus can get in. So you have a delivery vehicle for the virus wrapped in a lipid particle. So even in the resistant cell, you can mechanically deliver the virus inside the cell. Once it goes inside the cell, that resistant cell may support replication of the virus as well. So that's the important concept. Remember that. Susceptible cell means like it just can go in, it may or may not replicate. The same way the resistant cell, the virus cannot go in. But if you have a means to put the virus inside the cell, and this is artificial mean, remember that, not a natural means, then it may replicate or it may not replicate. Then the next one is a permissive cell has the capacity to replicate the virus, but it may or may not be susceptible. So what it means that the permissive cell may not have a receptor. It cannot go in. So a permissive cell has the ability to replicate the virus. So you can just think about a permissive cell can be susceptible or resistant, but it will replicate the virus, but it may or may not be susceptible. So just remember that that's what it is the capacity to replicate the virus. On the other hand, this is the important part that you are just bringing these two words together. A susceptible and the permissive cell is the only cell that take up the virus particle and replicate. Remember that we said that we can put a virus in a resistant cell. That's an artificial process, but in a natural process, without any means that you want to infect a cell line, without any lipofectamine, any lipid nanoparticle, anything like that, you just add the virus, it goes in the cell, replicates. You need a susceptible as well as a permissive cell line. So just make sure that just take your time to just understand this concept. You are going to get some critical thinking questions on this slide. So this is a very important concept. And if you have any questions, you can just ask me. I can repeat because this is going to be very important. All right, growing viruses. So routinely, we grow viruses propagated in the cultured cells. So this only in 1950s onwards, the cell culture, we could do the cell culture. Before that, the animals were before that, the viruses were only propagated through the animal models or the animals, basically. So you have to house these animals and they have a limited lifespan. You have to just inoculate new animals and it's a huge hassle. And then what happened is 1950s, when cell culture was developed, that was opened up a whole new door that you can just infect these cell culture and keep these petri dishes in the incubator and propagate the virus. But even at that time, there was some limitations in there. We are going to see that depending upon a continuous cell line and a permissive cell line, we're going to see that. Also, along with that, laboratory animals and embryonated chicken eggs were also used for virus propagation. And still we use that for commercial vaccine production, vaccine strain virus production. We're going to see it in a second. And also the plants, fungus, protists, and the bacteria, these were cultured in the laboratory to propagate the viruses. So these are the different ways that you can do that. And the funny part is that we are going to see that the plants now can grow recombinant vaccine strain animal viruses. And influenza viruses are actually being made in tobacco plants now as well. Not everywhere, but they are extracted from the tobacco leaves, recombinant vaccine strain influenza virus, and marketed. It is in the market. It's not any scientific, like a scientific fantasy or anything like that. So here we are talking about that once the cell culture came into the picture, basically, initially the cells were all what we call as a primary cell lines. So what is a primary cell line? If you have taken cell biology, it's easier. If you have not taken, then I'm just explaining that. So you take a tissue and then you just grind it up, mince it up, and then you mix it with the collagenase, trypsin collagenase. So that basically what will they do? That they will break up the cell attachment and the cells actually come loose. And then you just isolate those cells, a single type of cells. Remember that you cannot contaminate those with many different types of cell. It has to be a homogeneous one type of cell. You isolate these cells and put in a Petri plate in a defined growth medium along with some serum or you need some serum. Usually those are from the bovine calf serum. It contains the growth factors that is needed. So these cells will grow, but these primary cells, say for example, here you see that primary human foreskin fibroblast has a limited lifespan. You can, once the cells are growing, it just fills up the Petri dish or the flask. You take a little bit, put in a new one, it grows. That is a one generation. Then you take a little bit in the next one. Like this, each of these is called a passage of the cells. Once it is 5 to 20 times, these cells basically die off. They have a limited lifespan. Now you can see that this V38 human embryonic lung cell is a diploid cell strain, which can be passaged up to 100 times. It's a primary cell line, but it can be passaged up to 100 times and it's used for many of the vaccine productions as well. Now, on the other hand, what you are seeing that this mouse fibroblast cell line 3T3 and the human epithelial cells, the most famous one is a HeLa. These are continuous cell lines, means they are immortal. They are usually derived from a cancerous tissue or a tumor, and they have an infinite lifespan. So if we just talk about the most famous one is the HeLa. These cells were derived from a cervical tumor from an African-American patient. Her name was Henrietta Lacks. She went to Johns Hopkins, and the scientist, without telling her, removed or operated and removed the tissue, and they put it in the culture. And that is probably the most used cell line in the whole world. Is growing in every, like if you just go around the Clemson campus, probably 50 laboratories are having a HeLa cells. That's probably going to be the most common one, along with the other one. And all of them came from that one tumor, all throughout over the world. So it's kind of mind-boggling. So there is a fantastic book, if you have a chance to read that. Anybody read the book, The Immortal Light of, so The Immortal Light of Henrietta Lacks by Rebecca Skloot. Would read that book. It's really a great book. Immortal Life of Henrietta Lacks, Rebecca Skloot. You can just easily see that. in Amazon or somewhere else. So that's your HeLa cell line. So what is the problem of this continuous cell line? The continuous cell lines are aneuploid, means that they may have their chromosome numbers are not normal. They do not have the diploid number of chromosome. They may have something missing or something added, and they have a DNA sequence which do contain some of the sequences or oncogenic sequences. Basically, they're cancerous. So what happens that these cell lines are, you can propagate the virus, but sometimes they are not natural as natural cell. Like in primary cell have all the natural properties of a human cell. So some of these properties of the virus, when you are propagating them in these cells, continuous cell line, some of the properties may be compromised. So that's one of the limitations. The second is that many of the vaccines are now produced in these cell strain on the cell strains, but no vaccines are usually made in these cell lines. The reason is that suppose in the process, you are isolating the vaccine strain virus. Along with that, if there is some DNA, although you can do quality control on everything, but by chance, if you just isolate some DNA which contains the oncogenic sequence and you inject that vaccine, that oncogenic DNA can go into the human being and get integrated into the chromosome by some means, and that can lead to the cancer. So these are the things. So see what happens that when you have all these fears of the vaccine, this story is known by something. So if you take a cancer, some of these aneuploid virus and grow a vaccine against that, this can happen. But see, for the SARS-CoV-2, that's not the case. You are not getting any live virus, and they're not even grown in a cell. So that's what it is. So 9:15, we will stop at this virus cultivation, and we will finish the rest in the next class. So what you are going to see, that I will open up the chapter 1 quiz. You will see that under the assignment tab. You just go in there, you will see that, and I will open up the second one on Thursday. So you have 2 quizzes due Sunday, 11:59 p.m. All right, 10 questions. See you guys Thursday.